My biology role model is Dr. Kate Adamala. Dr. Adamala is an associate professor of genetics, cell biology, and development at the University of Minnesota. Dr. Adamala grew up with a fascination for science fiction. She received her Master's of Science in Synthetic Organic Chemistry from the University of Warsaw in Poland, and then worked with Pierre Luigi Luisi as well as Jack Szostak in grad school, where she studied RNA as well as the small peptide catalysis as well as liposomes. She completed her postdoc at Ed Boyden's Synthetic Neurobiology Lab at MIT. This is one of the reasons that I think Dr. Adamala is a good role model. She grew up with a dream and she made that dream come true. Dr. Adamala's hobbies, all I can say is one of them, and I can say from experience. She's a great baker. If you ever get the chance to try her red velvet chocolate chip cake, you can count yourself lucky. Dr. Adamala works in the field of synthetic cell development. The goal of this field is to create an artificial cell that has all the functions of a normal cell that we want and none of the functions that we don't want. Doing research on normal cells can be difficult, sometimes just due to the sheer complexity of the cell and everything inside of it, as we just saw in the last unit we learned. These simple cells can be used for metabolic engineering, drug development, and biosensing, according to the Biotech Institute. Simple cells also allow for collaboration around the world, where as long as you have the right ingredients and anyone with the proper knowledge could recreate a cell developed somewhere else. Dr. Adamala is also the coordinator for Build-A-Cell. Build-A-Cell is a community of scientists that focuses on the engineering of synthetic cells across the world. Build-A-Cell works to design synthetic cells that will eventually be specialized in industries such as pharmaceuticals or even chemical production. Here's an example of a specific experiment that Dr. Adamala worked on. In this experiment, as you can see in figure A, ribosomes were tagged with fluorescent protein. They were then encapsulated in an OA GMOA or an OA GMO containing vesicles. These vesicles are like little spheres around the fluorescent protein, around the ribosomes. In C, D, and E, as well as the images below, you can see the amount of cleavage that occurs in both types of vesicles, as well as E being no vesicles. And then the images below show that the gel electrophoresis results. Both the graphs and the gel images show that the vesicles support the survival and activity of the ribosome. And you can also see that D, the GMO version, supports it a little bit better. Dr. Adamala is quite rewarded. Here are just a few of her roles and awards. First off, Dr. Adamala is a McKnight Land Grant Associate Professor at the University of Minnesota. She's also a Polymath Fellow for the Geneva Center. This program awards a fellowship to 3 scholars who think outside of their field to avoid boxing science into distinct sections, rather than an ebb and flow of information. She's also the coordinator for Build-A-Cell, as I mentioned earlier, which is a collaborative group of scientists working to develop simple cells. On top of that, she's also the leader for SimLife, a biotech startup that focuses on the development of therapeutics through synthetic cell development. So, to wrap up, here's why I admire Dr. Adamala. She had a dream and stuck to it. She's incredibly hardworking, she's highly awarded, and she also works in a field that I have great interest in. Thank you for watching. Here's the sites to the images, as well as the citations.